At the end of this unit, we're mainly going to focus on cell division in eukaryotes. But before we get there, let's look at what happens in prokaryotes, organisms that are much simpler than eukaryotes. A reminder, a prokaryote is a bacterium. No nucleus, no membrane-bound organelles, very simple, very small. And cell division is the process in which we make two new cells from one parent cell. The parent cell is whichever cell we're starting with. It's your original cell. And when it divides, you end up with two identical clones of that parent cell, and those are called the daughter cells. Again, I just said this, prokaryotes are simpler. They don't have a nucleus. They don't have a space that holds the DNA, so it just kind of floats around. It's called a nucleoid. You can see in the picture to the left here, it's this purple area. Bacteria are the only things that are prokaryotes. And it's really important that you get this difference straight because it's going to come up all year long. Eukaryotes are more complex, have more DNA, have membrane-bound organelles, things like mitochondria, chloroplasts in plants, Golgi apparatuses, etc. Most eukaryotes are multicellular, but we do have a couple exceptions to the rule. Prokaryotes, though, can only ever be one cell. Is cell division, thinking about what I just said, simpler in prokaryotes or eukaryotes, and why? The answer is prokaryotes, and the reason why is because prokaryotes are simpler. They have less DNA, they're not very complex, and so cell division is going to be quite easy. The first step is that the DNA gets copied, and most DNA in prokaryotes is in a ring shape like this. So the DNA gets replicated and you end up with two identical copies. You can see in the picture here that it was copied in this first part, and then we ended up with two copies that go to the opposite sides of the cell. This is called chromosome segregation. The third step is that the chromosomes eventually completely separate, and you get two new cells as the plasma membrane grows and fills in that area. 3 simple steps. Eukaryotes have more DNA. So we just looked at prokaryotes for bacteria. We mentioned in the last video that DNA gets wound so it's easier to move in eukaryotes. Most of the time it's in that spaghetti phase, chromatin. Interphase is the phase where the cell is resting and DNA is most likely in that form. However, before the cell divides, absolutely everything needs to get copied, because if it didn't, your two cells that you would result in, your daughter cells, would not be identical. So DNA gets copied, organelles gets copied, and that's why you see down here, you have this one piece of DNA that gets wound up, and the chromosome, I kind of left this out last time in the last video, the chromosome actually has the two identical replicated pieces stuck together. A chromosome is technically this, it looks like a propeller, and it's still called a chromosome when it gets copied and you have an identical piece of the DNA held together at the center by a centromere. When the nucleus divides of a eukaryotic cell, it's called mitosis. So we copy the DNA, and then the nucleus goes through this division. The cytoplasm will then divide, and each new cell gets its own nucleus, and you end up with two cells. That's called cytokinesis. We'll go through these steps in more detail later. What you need to do next is complete the cell division worksheet and then move on to LO4.